So in this podcast, we're going to be looking into the structure and function of the membrane that surrounds the cell. So you've already looked at all of the organelles. We don't go into any much more detail on the organelles except for the cell membrane for very specific reasons, because we're going to talk about how things get across this cell membrane. And we're going to be doing a lab on that. And so we need to first figure out what is the structure and the function of the cell membrane. So here's a couple diagrams of a phospholipid that is contained within the cell membrane that you have already seen. We talked about these when we talked about the organic molecules. We talked about the phospholipids as different from the triglycerides in that this head region has a phosphate group and the glycerol group, and this tail region here contains those hydrocarbons. So we looked at the structure already of a phospholipid. Remember that we said that this head region here was a polar end, and the tail region was a non-polar end, which created these properties of the head regions being hydrophilic. Here's all of those heads here. And the tail regions on the inside here, these are the hydrophobic regions. We're going to talk a little bit more about what that looks like and how that creates a cell membrane in a few slides. I'm going to come back to a couple slides that look like this, but this is generally what the structure of a cell membrane will look like. Here you can see these large molecules here, the purple ones that are all sticking up inside and spanning the membrane. Some of them don't span the membrane. There's a couple here that are not spanning the membrane. These are proteins. Proteins are contained within the phospholipid bilayer. What you might also see here is that this is the inside of the cell. This is the outside of the cell. What you can see here, this is your cytoskeleton that basically anchors organelles to the cell membrane. We're also going to talk about a few other things. We're going to talk about these molecules here, cholesterol molecules. We're going to talk about these carbohydrate chains as well. But this is typically a structure of the phospholipid bilayer that I'm going to keep on coming back to. So in describing the functions of a cell membrane, I want you to think of the cell membrane as the outside of anything, in meaning that we can consider any outer surface a membrane. So for example, if we're considering our school, we can consider the outside of our school as a membrane, as a barrier that is provided to separate the inside from the outside. So in that sort of analogy, think of the cell membrane as its ability to provide a barrier separating the inside from the outside. What the cell membrane also does is it provides structure and support. So it provides structure in that it gives the cell its shape, as well as support because it allows things to move in and out of the cell. So it provides structure and support. And also, as I just mentioned, it regulates substances that enter and exit the cell. So regulates substances that enter or exit. Enter or exit the cell. Sorry, a little bit slow there. In this way, it acts as the gatekeeper. So it's just like our school. The doors, for example, would allow certain people, things, to enter or exit the cell. So when you're thinking about the cell membrane, don't be so scientific about it. Just think, okay, its function, it's the outside. What do all outside of any structure, what does the outside of any structure do? So here's how the cell membrane is usually described. It's described as a fluid mosaic model. Now, if you break down those terms, fluid is talking about the ability of things to move. How liquefied is something? And it's because of these phospholipids that are here that is actually quite a fluid membrane. And these phospholipids are constantly moving, allowing things to almost float along the top of the surface, allowing things to slightly shift within the fluid mosaic model. The mosaic piece to it is that we have proteins. These proteins here that are bobbing up and down throughout, moving side to side within this fluid bilayer. So this is usually described as a phospholipid fluid mosaic model. And so a typical test question would ask you to describe what that means. And you would state that it's a mosaic of protein molecules bobbing in a fluid bilayer of phospholipids. So let's come back to reviewing this phospholipid structure here that we called previously the dancing heads. So my first question would be, what is this made of? And if I just gave you this diagram, you can see what it's made of here. And here is this group that we would normally just have a circle around, and we just have a P in here. This is the phosphate group. So this is the head region of a phospholipid, and it is made of the phosphate group. Now that phosphate group, we call this the polar head. And these bottom pieces down here, the tail region, you can see here that they are made of hydrocarbons. So this is the tail, and it is the hydrocarbons, and we call these the nonpolar portion of a phospholipid. So these are nonpolar regions of the phospholipid. So if you remember back when we looked at the properties of water, and we realized that water is a polar molecule, and in the lab we did some experiments in which we showed that like dissolved like, that the like would mix with other like substances. So when this phospholipid is placed in water, what happens is that this nonpolar region right here, the tail region of the phospholipid, these are going to turn in. And so these are our hydrophobic areas. They are nonpolar. So this means water hating. So this doesn't want to be anywhere near water. Whereas in the head regions, these are hydrophilic. So hydrophilic. Philic means loving. So this loves water. So hydrophilic. And this means water loving. And so what happens when you place these phospholipids into any aqueous environment, in water, for example, the hydrophilic regions are going to stay on the outside of the cell's membranes. And the hydrophobic regions are going to turn in on themselves. This is what creates this bilayer, because these tails don't want to be anywhere near water, where these head regions are hydrophilic. They love being near water. And so therefore it creates these two layers. So here are our two layers. Here's our layer 1 of the bilayer. Here's our layer 2. And this is what creates this phospholipid bilayer, separating the outside of the cell from the inside of the cell. So we've looked at the structure of the phospholipid and talked about its hydrophilic regions here and its hydrophobic regions here that creates this bilayer. Well, we also need to talk about the other components that are within the plasma membrane. So looking at the proteins, and these are all these red ones, these red shapes here. Proteins can span the membrane, and some of them actually do not fully span the membrane but are just embedded in the membrane. So we have a few different ones listed here. For example, we could have a carrier protein, which would be, for example, to transport sodium and potassium across the membrane. Now that's really important in the nervous system. When we come to the nervous system, you're going to have to know that one very well. But here's another example. This one right here. This is a channel protein. And so what a channel protein does is it just allows certain ions or molecules to pass through. So in that way, the membrane is still acting as a gatekeeper, only allowing some molecules or ions to come through and some to exit. Another example here is these proteins here. These proteins we called glycoproteins, and here's the definition up here. Glycoproteins, here is a carbohydrate chain that's attached to this protein, and it will be used for cell recognition. We also have receptor proteins. Now this one would be a good example of a receptor protein in that it will receive a molecule or an ion and cause a secondary reaction to occur. So that's just some examples of functions of proteins that are very important within the cell membrane. So I just touched on this portion of the cell membrane right here, this carbohydrate portion. So what carbohydrates do, they allow the cell to be recognized as itself. So we've already talked about how in the ABO blood typing that all those people who have blood type A will have the same type of carbohydrate on the outside of their cell. And that's involved in an immune system response. If it deems that cell to not belong to the body, it will have an immune system response. So I'm going to break down, there's two different types of carbohydrates. And the next slide, I'm going to tell you what those two are. Okay, so there are two types of carbohydrates that we need to talk about, one of which is a glycolipid. Now, before I dissect down what that where that actually is, I've just drawn off to the side here an example. So glyco is referring to the carbohydrate. So if it's glyco with a lipid, it's a carbohydrate attached to a lipid. If it's glycoprotein, it's a carbohydrate attached to a protein. So here's an example of a glycolipid. You can see there's the carbohydrate chain, and it's attached to a phospholipid. So we would call that a glycolipid. Whereas a glycoprotein is the carbohydrate, glyco, attached to a protein, and that we call a glycoprotein. Remember that these are macromolecules, and therefore they are covalently bonded together. The last component of the cell membrane that I want to talk about is cholesterol. Now, normally, when we hear the term cholesterol, we have associated with being something bad because people now have high cholesterol levels, which can lead to clogged arteries and hence leads to strokes or heart attacks. But cholesterol is actually a main component, an essential component of the cell membrane. So I'm going to draw some circles around some cholesterol that is found within the cell membrane. So all these little yellow pieces here, this is cholesterol. So what cholesterol does is that it reduces the membrane's fluidity. Remember, we said that these phospholipids, they are a fluid mosaic model. So these are constantly moving. It is literally like pressing on the surface of, say, for example, if the water had a slight membrane, these would be able to move in that sort of manner. And so what these cholesterol molecules do is they stop the phospholipids from moving so freely to stiffen it up a little bit. And it also stops the membrane from becoming solid at room temperature because it maintains a certain fluidity. It doesn't allow it to be too fluid, but at the same time doesn't allow it to be solid. So those are the main components of a cell membrane. Let's do a quick review. So we've talked about the phospholipid and its hydrophilic head and its hydrophobic tails, and when placed in water, they turn in on themselves, creating that bilayer, 2 layers of phospholipids. We also talked about proteins. And proteins have many different functions. They act as carrier molecules, they act as transport molecules, and they also allow for certain molecules to be received by the cell and cause secondary reactions. We also talked about these carbohydrate chains, glycoproteins if they're attached to proteins, and glycolipids if they're attached to a lipid molecule that serve as the function to recognize the cell as self. The last one we talked about was cholesterol, and that cholesterol helps reduce the fluidity of those phospholipids so that the cell isn't too fluid, but also doesn't allow the cell to become solid at room temperature. So next we're going to be talking about how the cell membrane is selectively permeable, meaning that it doesn't allow everything to come in and out of the cell. It selects specific things, and a lot of it is because of these proteins that allow the selection process. So we call that selectively permeable. It only selects certain things to pass through, its permeability. So we're going to start talking about all types of transport that happen through the cell membrane after we finish our microscope lab.